There is a real-life evolution experiment happening right now inside of farms, hospitals, and even your own gut. And the result? Well, some bacteria are becoming almost impossible to treat. So today, I'm going to be breaking down the science behind the fight against antimicrobial resistance. This is real-world examples of the biology that you learn and need to know for GCSE and A-level biology. And don't forget that for your A-level, it's amazing to be able to know beyond the specification real-life examples to help with the essay, plus if you've got any interviews coming up. So if you do have any other interests in real-world examples that you want to know more about, then put it in the comments and I'll see if I can make some videos for you on it. So what is AMR? Antimicrobial resistance. When medicines such as antibiotics stop working to kill and therefore treat infections caused by bacteria, then you actually don't have any medicines you can use to kill that bacteria to treat the infection, and you're relying purely on your immune system alone. And this is antimicrobial resistance, or AMR. Now, for some people, that's not going to be much of an issue if you are fit, healthy, and you've got a really strong immune system. But for many people who might be very, very young and don't have a developed immune system yet, or much older and have a weaker immune system, or have any other condition that might compromise their immune system, this is incredibly dangerous and it could be life-threatening. Bacteria reproduce so rapidly that they evolve really, really quickly. And that's how, within my lifetime and within your lifetime, we have seen populations of bacteria becoming resistant to antibiotics, sometimes multiple different antibiotics. This is a huge, huge concern, but the APHA, or the Animal and Plant Health Agency, is one of the frontline organisations in the UK trying to fight, battle and prevent this AMR going any further. In particular, looking at it in animals, food chains and farming. And everything that we're going to be talking through and that they do links directly to what you learn for GCSE and A-level. So this is a real world example, therefore it could come up in your exam as an application question, but also it's useful for interviews and the essay for A-level. So let's go into the science then of antibiotic resistance and see how this links to what you learn. Starting with the fact that bacteria are prokaryotic organisms, and for both GCSE and A-level, you need to know the structure of a prokaryotic cell. So prokaryotic cells are much, much smaller than eukaryotic cells. They also have circular DNA rather than linear DNA. Some bacteria have plasmids. They don't have any membrane-bound organelles. So that means no nucleus, no mitochondria, no rough or smooth endoplasmic reticulum or Golgi apparatus. They do have a cell surface membrane and they have a cell wall, but the cell wall is made out of murin instead of cellulose, which is what we see in plants, a eukaryotic organism. Now, the structure of these cells is paramount to understanding how antibiotics work. There are different types of antibiotics and they target different parts of a bacteria cell to kill them or stop them from replicating. So one way that some antibiotics, for example penicillin, can kill bacteria is they prevent the cell wall from forming. And if the cell wall can't form, then the bacteria isn't going to survive. There are other antibiotics that prevent protein synthesis from happening. And if the bacteria can't produce new proteins, it's not going to be able to replicate. It's also not going to be able to survive. And finally, there are also some antibiotics that will prevent the bacterial DNA from replicating. And if we don't have any DNA replication, there won't be any binary fission and therefore no new bacteria will be produced. Now, this also leads into explaining why antibiotics do not destroy viruses. Viruses aren't living things. They are acellular, non-living. And all they're made up of is genetic material, a protein capsid layer around that genetic material, and then attachment proteins. So if you are infected with a virus, such as some of the viruses that cause common colds, and you take antibiotics to try and get rid of this cold, it will have no impact because some antibiotics affect cell walls. Viruses don't have cell walls. Some affect DNA replication. Viruses don't replicate the DNA themselves, they use the host cell that they infect to replicate their DNA. And protein synthesis, again, they aren't producing their own proteins, they are replicating inside of host cells, and they'll use the host cell's machinery to have their proteins produced. And this is why antibiotics do not destroy viruses. However, taking antibiotics when you have a viral infection has been one of the big causes in the increase in antimicrobial resistance. So how has antibiotic resistance developed? This is where we link to GCSE and A level, looking at mutations, natural selection, and evolution. Every time a bacteria divides, it's going through binary fission. That means we have to have DNA replication. We also have the cell getting bigger before the cytoplasm then splits and the cell membrane splits to get those two new cells. But sometimes when that DNA is replicating before the cell splits in two, spontaneous random mutations happen in the bacteria's DNA. Now, most mutations don't cause any changes because, A-level students, you know this, the genetic code is degenerate, meaning that more than one codon codes for the same amino acid. So even if there's a mutation, it might not change the final protein that gets made. But occasionally, mutations do randomly occur in the bacteria's DNA that result in the creation of a new allele, and this new allele provides them with a protein that means that they are now no longer going to be destroyed by the antibiotic. So that might be a change in the shape of a protein that is a binding site, or it might be it now creates an enzyme that's able to break down the antibiotic, or it might be able to create some protein that assists in pumping the antibiotic out of the cell. That's a whole range of other reasons. But the key here is suddenly, randomly, by chance, mutation has occurred. It now has a new allele that means this antibiotic will not kill this bacteria, and therefore this bacteria survives, whereas all the other bacteria you're infected with will die. This bacteria that now has that allele for antibiotic resistance has no competition for resources in your body. So not only is it surviving and replicating, it is able to replicate consistently because it has a constant supply of all the resources it needs with very limited competition. So the result is, after many generations of that bacteria dividing by binary fission, you now have a large population of bacteria that all have this allele for antibiotic resistance. And most bacteria divide every 20 minutes, so this happens rapidly. So therefore, we have had natural selection occurring, ending in evolution of this population of bacteria. So we've got this change in the allele frequency where most of the population is going to have this allele for antibiotic resistance. And this is your key example of directional selection for A-level biology. So what do APHA do then to try and battle this? They run national surveillance programs, which sounds quite complex, but it basically means they're constantly doing these things. They collect samples from farms and wildlife, they identify the bacteria at those sites, and they test which antibiotics they are sensitive or resistant to. So they will be using this aseptic technique to grow the bacteria that they find at the farms and on animals en masse, apply the different antibiotics and see which antibiotics are still effective at killing each of those bacteria. When APHA find resistant bacteria, especially ones resistant to antibiotics needed for human medicine, they act fast. That's because resistant strains can spread from animals and therefore food and then into humans. And that's why so much work is done looking at animals and food chains, because that is going to impact humans. For example, Salmonella, which causes food poisoning in humans. This is a zoonotic bacterium, meaning it spreads between animals and humans. So the link here is to GCSE and A levels that we have transmission and infection, there's food safety, pathogens, and immune response. And when resistant Salmonella appears, APHA's job is to stop it before it becomes established in UK farms. APHA is actually recognised globally as well by the World Organization for Animal Health as the only AMR accredited lab in the entire world, or at least as of 2025 when I'm filming this video, which shows you just how seriously the UK are taking antimicrobial resistance. So I said that they are testing to try and see, are there resistant strains? But not only that, they're trying to then stop those strains from spreading. So the sorts of things they're researching to try and help with this are disinfectants that might be able to kill these strains, bacteriophages, which are basically viruses that kill bacteria, and also rapid AMR detection tools to locate resistant bacteria far more quickly to try and destroy them before they are able to spread. So why does this matter? Obviously, I'm linking this to your GCSE and A level, but it matters beyond your learning for your exams. AMR is literally an evolution example happening now. So there we have it. We have linked a real world example. It is Antimicrobial Resistance Awareness Week, or at least it was when I was filming this in the middle of November. And it is so important that you do have this awareness that you are not misusing antibiotics. You shouldn't be taking them unless they're prescribed, and it should only be for bacterial infections if your own immune system is too weak to destroy the bacteria yourself. You should always take the full course of antibiotics that you're given as well. So make sure you're doing your bit to try and protect the most vulnerable in our population. Plus, this links perfectly for so many essay titles that could be linked to bacteria, prokaryotic cells, immunity, gene technology, natural selection, and evolution. If you found this helpful and you want more of these real-world examples that could link to essays, but also show you how your GCSE and A level is relevant in the real world, and maybe prepare you for application questions, then let me know in the comments.